My name is Duncan Ross. And today I'm going to speak about lab-grown virgins, vampires, exosomes, and parabiosis. This is one of my favorite movies down there. So in history, we've been speaking about vampires for quite a long time. There was Ambrosio in Greek mythology. And so it makes me wonder if over thousands of years ago we were discussing parabiosis and living forever, what were people doing? What were they eating? Or who were they eating? It's kind of an interesting thought. So what is the key to immortality? So can we do this? People say that, Dr. DeGray said that the key to living 1,000 years, or someone who is going to live 1,000 years has already been born. Is that here or not? And I believe it actually is. So I think that we are suppressing aging now with what I'm going to discuss, which is placental mesenchymal stem cell derived exosomes. I think the blueprints to reverse aging have already been published, in fact, in Nature. A method to rewind aging, which is very, very strong, was published about a year ago. And it's a great time to be alive, although I'm not sure CRISPR is what's going to handle it because of off-target effects in the genome. So what I'm proposing is a shotgun approach. If you think about the fact that cells have been cultured in fetal bovine serum for 50, 60 years, we don't even know exactly what the factors in fetal bovine serum often are causing cells to grow. I'm going to propose that it is a number of proteins, and more importantly, messenger RNA, that will do this. In my case, I've given myself the treatment that we're going to see, which is 300 cc's of exosomes derived from placental exosomes. And 8 months later, I could see the wrinkles on my forehead go away. So it's kind of interesting. That was without using cream. So my background is in bone marrow transplantation for leukemias. And bone marrow transplantation we discussed today was created in the 50s, or started in the 50s. So much of the information about controlling inflammation has been known for quite a long period of time. So in the regenerative medicine space, it's interesting to think that we think that we're discovering treatments every day. But in fact, if you just hung out with a hematologist for a while, you would probably know a lot of these answers. So the way we do transplantation is by taking cells from one adult and giving it to another adult. But that treatment always requires immunosuppression of the recipient in order to avoid allogeneic rejection. In a lot of the cellular transplantation we're talking about today in regenerative medicine, there is no suppression of the recipient. Therefore, those cells which are being transplanted in the case of umbilical cord blood will be rejected within hours, not days, hours. And that was the basis of my PhD, was tracking stem cells that I had labeled from one mouse into the next one. So in the 50s, at the same time that they're developing bone marrow transplantation, there were other experiments about parabiosis. And that would be taking two mice, a young mouse and an old mouse, hooking up their circulatory system and observing the effects. And as many of you know, the young mouse got older, the old mouse got younger, but the old mouse improved its ability to run through a maze, which is very interesting. So in those two mice experiments, which have now been resurrected in many centers such as Stanford and others, it's been found that the plasma alone had the same effects as exchanging the whole blood. So you think about what's in blood. A lot of people, when they tell you you can use cord blood for regenerative medicine, they say, well, there's stem cells in it. But there are no mesenchymal stem cells in cord blood. I do believe that transferring plasma between two mice works. And in fact, in a lot of the things we're probably talking about here today, there are people that say that, oh, this is just another huckster cycle. And this particular person I thought I would call out because he's an MD, and he's a clinical neurologist, and he runs his popular science podcast, The Skeptic's Guide to the Universe. Well, everything here, if it's working, even if you have anecdotal evidence, there must be a reason. So I never like to look at what we're discussing in regenerative medicine and say it's wrong. I just want to know exactly what it is that's causing that regenerative result. So this is one of the companies doing young blood to old patients. It's called Ambrosia. This is another company called Alevion. And this company isolated protein factors that they found that were highly expressed in the younger blood and in the younger mice. One of those proteins was GDF11, growth differentiation factor 11. So obviously, if you're finding that GDF11 is overexpressed in the younger mice, you think, well, I've got the holy grail, I've got the secret sauce. I'm going to express that one protein recombinantly, and I'm going to apply it to the patient, and they should get younger. Well, that's not how it turns out. If you give GDF11, nothing happens. And why does nothing happen? Because a protein is very labile. Yes, proteins are the end effectors in cellular work, but they're not really the longest lasting. And there's a reason for that. If we have to change directions quickly, we need proteins that are going to denature quickly so that we can express them again when we need them and they don't hang out for too long a period of time. If you have a protein that's telling you to upregulate something and it sits around for 2 weeks, you're going to have overgrowth and it's going to be very dangerous. So the labile nature of proteins is by design, and it's very important. So what then is the fount of youth? What is the center of our body? The center of our body is the mesenchymal stem cell. And the mesenchymal stem cells are throughout your body. They're disseminated, and they control growth. They especially control growth when you're growing. Because if you're going to become a tubular epithelial cell, you're not telling other cells to grow. So something must be controlling the growth cycle and the chemokine gradient between cells that are growing within a body. So where do these cells occur? I just told you that there are no mesenchymal stem cells in the blood. But mesenchymal stem cells are correlated with the blood. They are termed parasites. They are attached to venules and arteries, and they secrete these growth factors into the blood. Therefore, in the experiment that I just showed you, where the blood cells were removed and only the plasma given, you can therefore, correlate the expression of growth factors into the blood from these mesenchymal stem cells to what's happening. Therefore, you don't need the cells. Well, so what are they? Well, we know that stem cells, when placed into a wound, are going to improve the timing of wound healing. We also know that allogeneic mesenchymal stem cells, when given to a chronic graft versus host disease patient, will suppress GVHD, and that's about 14 million cells in a 70-kilogram patient. If you take out bone marrow for regenerative medicine, there are only about 30,000 mesenchymal stem cells. If you take out fat, there are about 150,000 mesenchymal stem cells per 60 cc's of fat. But if you should give 14 million cells, which is quite a large number, you can suppress chronic graft versus host disease. So the disadvantage of cells is that if you've taken them out of their niche, their cell persistence is now going to be very limited. You've removed them from the vasculature that's feeding them. What's going to feed them? They die. If you're going to create 14 million autologous mesenchymal stem cells, it's going to take you 6 weeks to express those cells. Third-party cells, as we know, are also lead to rejection because they are not immune privileged. And cells are not, in fact, FDA approved unless they have an IND. So if these cells are going to be rejected because, in fact, they're immune evasive, they're not immune privileged, and they're immune evasive because mesenchymal stem cells don't really carry MHC class II on their cell surface, so they don't get rejected. But if you could keep a cell and the cell factors that that cell makes, what kind of cell would you want to get it from? You would want to get it from the youngest cell that you could get it from. So we have two kinds of virgins, two kinds of cells we can work with, but what is the best reservoir of cells to work with? So an adult mesenchymal stem cell secretes about 110 factors, and I'm going to show you what some of those are. They can be growth factors, they're anti-inflammatory factors. In fact, they're tumor-suppressing factors. Whatever a young cell secretes equates to about 300 growth factors. And think about what a young cell is. A young cell is not tumorigenic. A young cell is very anti-inflammatory. If you cut a fetus within the first trimester, it will not scar. We scar very readily, right? That's because of an overactive inflammation. So we have these 200 to 300 proteins that we would like to get to instantiate growth. Well, in the past couple of years, there has been an explosion of the understanding of a type of molecule that is secreted by the mesenchymal stem cell. And in fact, it's not a molecule. It's called an exosome. And an exosome is a membrane-encapsulated lipid vesicle of about 100 nanometers in size, up to 400 nanometers in size, and down to 30 nanometers in size. This lipid vesicle surrounds proteins and, more importantly, messenger RNA. And why is the messenger RNA more important? Because from one messenger RNA, we can secrete thousands of proteins or translate thousands of proteins. So in fact, an MSC in the body, and this is how cellular communication works, an MSC secretes an exosome, which goes into the neighboring cell and allows that neighboring cell to express proteins that it didn't even know it wanted to express. It could be an IGF-1 receptor molecule. It could be an antiviral molecule, an IL-1 alpha, for instance. It can be anything that mesenchymal stem cell is telling that cell to create without having to involve the DNA of that particular cell, because the messenger RNA is now being expressed in the cytoplasm. So don't think about exosomes as small cells. I have not had a cellular product in my FDA-registered and inspected laboratory for the last 5 years. There's no purpose to sell a cell or use a cell, as I said, because of the allogeneic rejection issue. But I do want those messenger RNA. I do want microRNA that can control the expression of other genes, and I do want proteins. This is what an exosome looks like. It's a small vesicle which is budding out of a larger cell. So what is the benefit here? The benefit here is if you're growing cells, autologous cells for that patient that I said we were going to treat, you would be very, very worried that that cell culture would become contaminated. There's no way, it's very difficult to get bacteria out of a contaminated cell culture. But in the case of exosomes, you can sterile filter them because no bacteria is the size of an exosome. You can also easily test them for sterility and endotoxin levels. They can be concentrated, and they cannot be rejected because they are too small. And an exosome, in the case of one coming out of a mesenchymal stem cell, does not contain HLA or specifically MHC class II that we were just talking about. That's not the same for all exosomes. Every cell you can think of secretes exosomes. So you might imagine, well, there are more anti-inflammatory exosomes, maybe from regulatory T cells. There are inflammatory exosomes. An exosome is simply a method to carry a message around the body. These can also be frozen normally. They don't have to be cryopreserved because they're not cells and they don't have to live. So in fact, because you can concentrate them and control them, you can make an exosome more effective than a cell could be by concentrating the amount of cell factors that are secreted by that cell in X period of time. So I could take 25 days worth of mesenchymal stem cell secretions and concentrate them into one product instead of having one cell that maybe will persist for 7 days. So in Halloween of 2014, I had a fortunate experiment. I had seen that the growth factors from a younger cell could make an older cell grow in culture. I had a 70-year-old COPD patient, and I couldn't get her cells to grow. So by simply transferring the exosomes from the younger cell to the older cell, those cells started to grow. So immediately I knew I had in my hands something that could be used therapeutically. So I was at a friend's house. He threw water on a gasoline fire, and it burned his face. So this is the second-degree burns from his face from throwing water on the gasoline fire. So I went to the freezer, and I took out 250 cc's of these exosomes, and I gave it to him, and I said, spray it on your face 3 times a day for 7 days. And so on day 1, you can see he's not very happy there. But on day 7, he's looking pretty good, and by 6 months out, he has a new skin, and not just a new older man's skin, an older younger man's skin, an older baby's skin. And in fact, there are 30 types of collagen. If you look right above his eye, there's 30 types of collagen in that skin, where there's only 8 types of collagen in the skin that you see between his eyes up above on his forehead. So that was a really seeing is believing moment that, you know, it's really the younger cells which are accomplishing what we want to accomplish, and the younger cells that are going to make us grow in the fashion we want. Now, we don't want to overgrow, right? So one way I like to think about this for everyone is by lactose tolerance. We are all lactose tolerant when we're younger, but we're not supposed to be lactose tolerant. It's only those from Northern Europe and us that are really lactose tolerant. That happens to a lot of growth factors. That's how the body works. That's how the body keeps us from overgrowing. So in an old person, the lactase enzyme is covered over in heterochromatin. And if we don't open the DNA somehow, we can't access it to express that protein. But in younger animals, we have open histones, which are controlled by histone deacetylases or histone acetylase transferase. And in that fashion, we can open the gene and we can express it. So theoretically, you should be able to become lactose tolerant again. I haven't shown that with the exosome yet, but that's also what's happening to other growth factors like insulin growth factor 1, VEGF. We get old because our DNA covers over. Fortunately, we're able to pass it along to our children who, again, after millions and millions of years, can grow again anew. So like I said, we really want to stop this growth lest we become Andre the Giant. But we would like to open it up again for a short period of time. So remember, we go from DNA to messenger RNA to protein. The problem is proteins degrade, and what about the speed with which they degrade? Have you ever really thought about that? Well, IGF-1 and VEGF degrade very rapidly because, like I said, you wouldn't want to be causing vasculature, overgrowth of vasculature. You wouldn't want to be causing over tissue growth. But the half-life of the VEGF messenger RNA is over 8.5 hours. The half-life of IGF-1 messenger RNA is 4 hours. And in the rat, it has been shown that some messenger RNA can persist for a period of up to a month. So by simply giving the message as a messenger RNA, you can express thousands of the protein of interest without having to actually have the protein there or be recombinant or be overexpressed. So this is what ribosomes on a messenger RNA actually look like expressing proteins. This should really give you the idea that one messenger RNA can make thousands, and in fact, it makes them all at the same time. The ribosomes jump onto the messenger RNA, they run down it, and they express. So within a 4 or 8 hour period of time, you can make lots and lots and lots of protein. If you just gave one protein that you made, it would start degrading right away and it would be gone. So this just shows that messenger RNA have different kinds of stabilities. Some of them are unstable so that they won't cause too much growth. Some of them are very stable, so they'll be around for long periods of time. In the exosomes that we create at Chimera Labs, we can run growth factor panels on them and see what we actually have. So this is one that I did here. On the right side in the red, you see the exosomes that we make. And I can see very clearly what the product is going to do. I have 3 products here. I have room temperature amnio over on the side. That was what somebody gave me. It's impossible to have room temperature amniotic fluid, but I had to prove it, so I ran this panel on it. In the middle, you have an amniotic fluid product that we also make. And on the right, you have the exosome product. In the exosome product, you can see high levels of VEGF. You can see TGF-beta3, which is the holy grail of anti-inflammatory proteins. If you take TGF-beta3 and put it in a dish with an inflammatory T cell, it will turn that T cell into a non-inflammatory T cell permanently. So if you're arthritic, that T cell, which was specific for that antigen, now will still be specific for it, but when it sees that antigen, it will express anti-inflammatory factors. In fact, you tolerize the cell. So the same thing goes for the messenger RNA now. If I see those proteins in my product and I know they're going to disappear rapidly, I should be able to show that I have the messenger RNA as well. And indeed I do. You can see TGF-beta3 here. You can see VEGF. You can see stem cell factor. All these messenger RNA are shown to cause growth. And interestingly, just like that experiment we were showing before with the parabiosis, we have the messenger RNA for growth differentiation factor 11, the fountain of youth protein. And these are other, here I show we do have messenger RNA for telomerase and histone acetyltransferases that I was discussing, which open the DNA, which allow the expression of lactase, which allow you to eat milk. So the difference between what we're doing at Chimera and what other people may start to do is that there is a tissue marketplace where tissue is taken from the mother and sold right away. That brings a lot of problems into it. You don't know, without testing
that specific donor, where they could have Zika, they could have Epstein-Barr virus. You are obligated to test for HIV and other pathogens, but you don't know a lot about that specific donor. You don't even know if that donor is good. So just as with lab-grown meat, they're growing meat cells in the laboratory to create a, derived from animal, but not really from a dead animal beef. We take one donor, and in fact, in the last 3 years, I've used the same little girl to treat all the patients that I've treated. So this is some before and afters really quick. On the left, you can see before. On the left, you can see 2 days after. That's by applying these baby exosomes with their baby collagen to the patient. This is the same thing. You cause a little inflammation to cause cells to come running in. You treat them, and then that's the result. The same occurs in hair. If you do some Googling of stem cell factor in hair, you'll find a number of publications that discuss exosomes and the treatment of hair. And in fact, we are seeing good results. This last one, sadly, I can't show you. This is an MRI of a paraplegic patient who can now do squats and walk by treatment of exosomes intrathecally. These exosomes that we're making, as I said, you can control their sterility, you can control their concentration, you can tell you how many numbers they are, and I test them down to intrathecal endotoxin levels such that they're safe being injected in the central nervous system. And we have treated thousands of patients at this point, probably about 100 spinal cord injury patients. So that's all we got for today. I have a much larger talk. If you want to hear it, you can check out the webinar, but I didn't want to get too sciencey with you today. If you have any questions, I'll be here.